and welcome to this week's TBS News Digest. I'm your host Niharika Singh, and I'll be bringing the latest scientific news from the wondrous world of biology to you. So let's begin. In a rare and astounding event, a group of shark hunters in South Africa accidentally rediscovered a coelacanth, that is a fossil fish which was declared extinct in 1938. Coelacanths can be traced back 420 million years ago and are believed to have existed even before the advent of dinosaurs on the planet. Anatomically, coelacanths consist of 8 fins, large eyes, and a small mouth, and they also have white spots which act as a distinguishing feature. Researchers now want to implement conservative measures to protect the fish. Interestingly, it is believed that the undersea canyons in Madagascar might become the Indian Ocean epicenter for coelacanths. Next in line, 2 Cambridge University professors, Shankar Balasubramanian and David Teneman, who developed a super-fast DNA sequencing technique, won the Finland's version of the Nobel Prize. Their sequencing technique, that paved the way for revolutionary healthcare advances, won the €1 million Millennium Technology Prize. 20 years ago, the first attempt to read the sequence of 3.2 billion letters that makes up the human genome took a decade and cost over $1 billion. Thanks to next-generation sequencing, the process can now be performed in one day for just $1,000. And the technology is used over 1 million times a year, most recently to track coronavirus mutations during the pandemic. NGS is now widely used in the diagnosis and treatment of some cancers and rare diseases. Next up, Professor Rohan De Silva's lab at UCL had recently published a paper suggesting that the role of an antisense long non-coding RNA in the production of major Alzheimer's disease-related protein tau. This protein, known to its cytoskeleton stability, is known to be overexpressed in Alzheimer's, at which point it becomes toxic to the cell, eventually leading to dementia-like symptoms commonly associated with the disease. By identifying the regulating mechanism of this protein, the scientists now wish to use treatments to try and prevent the overexpression of this protein. They also claim similar gene-regulating mechanisms for other proteins like alpha-synuclein involved in Parkinson's disease. Moving on, a study shows that bacteria colonize our gut during and after birth. The study was done by Dr. Thorsten Braun, Catherine Kennedy, and a multi-institutional team that studied meconium samples collected from 20 babies during breech cesarean delivery, putting to end the theory that humans are colonized by bacteria before birth. This key first step in our understanding of gut bacteria could help stage possible interventions for only good microbiota to colonize the gut of babies. Next up, scientists belonging to the Oregon State University are closer to finding a gene therapy method to enable the deaf to hear. The researchers suggested that otoferin, the molecule responsible for the regulation of neurotransmitters from sensory cells, requires the transmembrane domain along with its functional domain if it is to be used in gene therapy. The transmembrane domain of this protein was found to aid in the tethering of vesicles for neurotransmitter release and has also shown to aid in the maturation of the sensory cells. Thus, the transmembrane domain should be a part of any gene therapy for the defect. Next up, a new study published in the Bioscience Journal evaluated the similarities and differences between human epidemics and biological invasions by plants and animals. The authors suggest that the fundamental concept of invasion biology, which is the invasion of biological species into new environmental niches, can help study the different stages of epidemics. The fusion of invasion biology and epidemiological study can help us to upgrade prediction, design efficient prevention and treatment protocols, elevation of invasion species, and elimination of infectious disease outbreaks such as epidemics and pandemics. Sargassum bloom, a floating brown seaweed, turns into a toxic dead zone due to rise in nitrogen from natural and anthropogenic sources. Studies led by Florida Atlantic University shows a 35% increase of tissue nitrogen. Since the 1980s, harmful sargassum blooms will have catastrophic impact on coastal ecosystems, economies, and human health, researchers say. The Great Atlantic Sargassum Belt since 2011 has extended up to 8,850 kilometers, supported by increased nitrogen inputs from sources such as terrestrial runoff, nitrogen fixation, anthropogenic emissions of nitrogen oxides, biomass burning, and production of synthetic fertilizer, nitrogen. Human activities like population growth and land use changes have increased nitrogen pollution at high risk and above a safe planetary boundary and degradation of estuaries and coastal waters since at least the 1950s. Now let's move to evolutionary biology. A study by the University of Oxford and University of Birmingham proposes that the reason for the blooming of placental mammals like you and me during the Cenozoic era is not because of the relaxation of constraints after the extinction of non-avian dinosaurs, but rather because of the extinction of non-therian mammals. The authors analyzed morphological features of various species to conclude that the reason modern mammals did not spread and evolve was because of the competition from other now extinct mammals rather than the dinosaurs themselves. Finally, on May 17, 2021, the famous Darwin's Ark, New Darwin's Island, in the Galapagos archipelago had its top part removed because of erosion and now stands as just two pillars. The Ark region, a common tourist spot, also seems to be important for various aquatic species like the whale sharks. So important is this landmark. One tourist company calls what remains of the Ark the pillars of evolution. So thank you for watching. If you learned some good stuff from the video, then go like, share, comment, and also subscribe to our channel, The Biology Society.